Okay, good afternoon everyone, and welcome to the LSTM seminar series. So our speaker today is very recently appointed, so thank you very much to Eva for agreeing to give a presentation so quickly after her appointment in January. So Eva Heinz is taking up a position of lectureship in the in LSTM, having come from a background which included an MSc and PhD at the University of Vienna, before she moved on to postdoctoral fellowship positions at the University of Newcastle, which is in the northeast of England rather than Australia, and then at Monash University, which is Australia rather than northeast of England, where she studied topics such as bacterial cell surface evolution and genome evolution of eukaryotic parasites. And then more recently, she's been working at the Wellcome Trust Sanger Institute. And her work there is very much the topic of what she's speaking about today. She has many ongoing collaborations still going, so I think it's work sort of completed and in process, much of it unpublished, so we're very, very lucky to be hearing this today. So thank you again to Eva. We look forward to hearing your presentation. Thank you.
Okay, thanks a lot, David, for the nice introduction. And yeah, so today I'll tell you something about kind of like the main organism I've been working over the past couple of years, I guess, which is kind of a representative of a phenomenon really that we're seeing kind of like lately, and that's Klebsiella pneumonia, whom you've probably heard about at least in one or the other news report. And so really, what is Klebsiella? So this is what it looks like, which is also a bit of a challenge when working with it in the lab, which I had the pleasure to do for a while. It's got quite a big capsule, and that really is also one of its main virulence factors. So we kind of like know that it's very good at hiding from the immune system. And when you grow it on a plate, it's very easily recognized. It looks a little bit like somebody sneezed on your plate. So you've got these kind of like huge mucoid colonies. It's a gram-negative bacterium and a member of the Enterobacteriaceae, which have recently been extremely good just in kind of like collecting antimicrobial resistances. There are kind of like a lot of similarities, especially kind of like just on a day-to-day work to E. coli. It does have a large polysaccharide capsule, and without the capsule, we do know it's avirulent. So if we would get rid of that, you can't find any kind of like isolates that don't have any form of polysaccharide capsule. And it's an environmental organism found sort of like everywhere. We don't really know where, but sort of in the environment. And we know that it's also part of gastrointestinal carriage. So it's not necessarily a pathogen as such, like salmonella typhi. Usually when you catch that, you've got a problem. Whereas for Klebsiella, it's not necessarily actually a pathogen as such, which is part of the problem, sort of. It's actually a species complex, so a lot of work has been done over the couple of last years only to actually really identify it. This tree is based on a kind of like the major study which started the whole Klebsiella genomics field from Cat Holt. The majority of clinical isolates are definitely Klebsiella pneumonia sensu stricto, and we also have two subspecies in the complex which always come along as clinical isolates. Again, we don't really understand kind of like the difference between those two well. There is some plant association here, but you can also find it in clinical isolates. So this is all quite, we don't really know, for example, what the difference between those three guys are. And just, I think last month, Patrick Brice from the Institut Pasteur has published, so two more subspecies have been found. And yeah, I think I've gone through most of these points already. This is kind of like the main challenge really that I think we are facing with Klebsiella, and this is when you look through kind of like the last century, and when you just look in PubMed for the kind of like title term and what sort of publications you get. And so a lot of my colleagues work on typhi or cholera, which I think is completely unfair, because if you look throughout the century, sort of there's a huge body of kind of like research that has happened on these what I call like proper pathogens, so where we know it causes a disease and kind of like we know something about the epidemiology. Whereas Klebsiella and also Acinetobacter and Enterobacter, these are really all these kind of like recent gram-negative organisms which have become resistant. It's kind of like dumpling along here, and then here suddenly in the last few years we really have this explosion in literature, which is unfortunately I think not directly related to how much we actually understand about the organism. And this is kind of like really the question. So like, how did we go from here? So this fantastic finding from Alexander Fleming, who got the Nobel Prize 1945 for this, for penicillin, down to here, sort of like the antimicrobial apocalypse, like it's framed in the public media. Yeah, and so what we know for Klebsiella, that's definitely kind of like the case, is it is extremely good at acquiring resistances. And so acquired resistance in bacteria, it's usually sitting on a plasmid, and then you acquire the plasmid, and with it genes that help you to become resistant. And what we see here is a very precious collection published by Kate Baker, who is kind of like next building. The Murray collection, which is from the pre-antibiotic era, so before they were exposed to all these antimicrobials. And these are these little blocks here, and just in red, these are all acquired resistance genes. And you see we have this nice kind of like empty blocks wherever I've boxed them without. And then in the recent years, this is actually still what I've compared it to, quite a friendly collection as such. All these acquired resistance genes. So we kind of like know part of the problem of Klebsiella, or it looks like it. And it really is a global picture. The surveillance data at the moment, so there's a lot of effort fortunately being put into that. So this was the first report from the WHO, which was from really different kind of like settings and data points. So this is kind of like to be taken with a lot of caution. But just to give you a kind of like general picture, so it is kind of like spread all over the world. With varying percentages of severity, but again, this is between sort of like nationwide surveillance schemes, and other countries have just submitted hospital data. So this is at the moment a very scattered picture. We can not rely on too much, sort of. Just before I jump into, so the kind of like the rest of the talk, I guess, will be about kind of like antibiotic resistance and what can we do about it or against it, more like. I also have a little bit of work on kind of like the alternatives, and so kind of like here I've put as a question like, what about our immune system? Can we actually make that work a bit better? And so I've got an ongoing collaboration with Peter Taylor at UCL, who has kind of like spent most of his scientific life on complement resistance. And we've got kind of like some studies in Klebsiella and Acinetobacter ongoing. Unfortunately, again, there is no easy answer, like quite often in Klebsiella, so it's not as often publicized that a certain capsule type protects you from serum. And also a very nice collaboration I had with Alex Bateman, and which I'm kind of like continuously working on, is to identify vaccine targets or monoclonal antibody treatment targets to kind of like avoid the whole antibiotic resistance problem from the start. So just to kind of like follow up on that, why don't we just target the capsule? Because I said all Klebsiellas have to have a capsule, and it's kind of like very prominent. This is kind of the summary of the answer. So these are the, this was from 2016. This has grown in the meantime again. These are the different capsule loci we find in your standard Klebsiella collection. So almost every kind of like strain or lineage we find has a different capsule profile. And they also keep evolving. So this is the highest, the spot in the genome, which is the highest recombination rate by far. So this is just kind of like obviously kind of like likely a result from immune pressure, but also pressure in the environment, for example, by being targeted by, as we've got here up here, bacteriophages, which might actually be a main driver of Klebsiella evolution in the environment or in its more natural habitat. And so this is some work that was actually really predominantly driven. So this is the sort of like team capsule from Kat Holt at the University of Melbourne, who has done a lot of research in Klebsiella in general, and Kelly Wires and Ryan Wick. And this also ended up in kind of like an online kind of like platform where you can submit your genomes. And this is also interesting to follow the epidemiology, for example, because we can spot, for example, switches in the capsule type and then follow the lineages more. But yeah, obviously as an antigen target, this is unfortunately not very useful, sort of. And so the first study I want to go into more detail was really led by Hassan Ejaz from Pakistan, from the Children's Hospital in Lahore. And this is so this hospital's got a primary catchment area of about 10 million, and the wider area, where it's the only tertiary children's hospital, is 100 million people. So this is a huge kind of like setting. And at the time the study was done, there was no computer, so this is based on paper records and just very challenging conditions. And they have a continuous problem with Klebsiella. So this is not what we see quite often in the literature, an outbreak, but just a continuous sampling from diagnostics, from bacterial infections. And so Hassan really did this monster effort, starting with over 40,000 samples of presumed bacterial infections, and then selecting for Klebsiella and what was the main issue at the time were ESBL, so that is extended spectrum beta-lactamase. These are enzymes that the bacteria carry, so this is kind of like the nomenclature we use quite often, ESBL, and those are the kind of like target the third generation cephalosporins, which was a main or like one of the major kind of like hits in the antimicrobial resistance, because cephalosporins or beta-lactams in general are very good drugs. You can give them to a lot of patients, you can give them to kids and so on. You can kind of like give them by oral treatment. And so the other point, and if you don't remember much else from my talk, then just kind of like keep this in mind. Klebsiella really causes high mortality rates, and that makes it different from, for example, E. coli. And so here we've got it in neonates, where if you combine death and left against medical advice, which according to Hassan was mostly people just taking the baby out so it doesn't die in the hospital, we are at over 50% negative outcome rate. So this is a huge problem in the children's, and this is a continuous problem that they've had, so not a recent kind of like or a temporal situation. And so first of all, Hassan really did a monster effort on kind of like collecting all sorts of patient data, so jaundice, fever, cough, kind of like all sorts of metadata, length of hospital stay, outcomes, or mortality. Nothing is associated with anything. So here on the side, I've plotted the different wards and also the patient age, and these are kind of like our different Klebsiella lineages, and here I've got the major sequence types, and really nothing correlates with anything. So there's nothing that kind of like makes like a lineage more prone to a certain type of disease or so, at least as far as we can see at this stage. And here then, when we look again at kind of like virulence, which is not necessarily virulence as such, but being able to survive better, so this is mostly siderophores to steal kind of like ions better from a host environment. And then in red again, I've got the acquired antimicrobial resistance genes, and in blue the plasmid replicons, which is kind of like a sort of like a parallel, like a 16S typing scheme for plasmids. So you get a rough idea of what sort of plasmids you've got. And again, you can see that this is not like one lineage or one resistance type that just exploded throughout kind of like the population, but we've got this huge pot of diversity with some lineages, kind of like, for example, here, being very successful, and then some just coming in and out, just being like one or two isolates. And they're all equally resistant, so they were all pre-selected for being ESBL carriers. And so when I wanted to put it kind of like in a bit of a global context and see where our lineages fall to, so at that time there weren't that many published studies, so I've compared it to our global collection. There was, quite interestingly, at the same time, an outbreak in Nepal, which was led by Steve Baker, the investigation. And then there was a data set I got from Spain where they have quite a problem with oxagenes. So these are beta-lactamases. And so when I put it into, so if you just look at the inner ring of the tree, and so these four colors are the four studies I've just mentioned. So what you can see, so the one thing about Klebsiella is really that everything is everywhere. So we don't have a lineage that is specific for any kind of like any location. You can see that there are definitely biases. So for example, this was actually the major lineage in our setting, which in all other data sets I've looked at so far comes up as one or two isolates. And then, for example, here you can see, so I've taken a closer look at that. In green, we've got the Nepal outbreak, and in yellow, the Pakistan. And so we do see kind of like similar lineages in the same area, but we also see this huge diversity kind of like floating around, but making it into the clinic, into a patient isolate. And so when I've compared this kind of like this little part in the tree up here just, and zoom in on that a bit more, again here you've got the isolates from Nepal, from the same sequence type as the ones from Pakistan. And again, you see, even though it's the same area, they've got a completely different resistance repertoire, as well as virulence plasmids. So again, this is not kind of like one lineage which picks something up, like you sometimes see, for example, in Salmonella typhi, H58, and then just expands across the area. But there's a huge diversity and mobility of elements going in and out of our Klebsiella population, basically. And one of the questions that Hassan had is also that they saw a change in their diagnostics. And so they usually do kind of like counter screening with cefoxitin, which is used to distinguish AMP-C isolates. And they've seen kind of like not a huge, but a gradual increase in isolates, which also had this phenotype. And so of course one of the question was, is there just a new plasmid or a new lineage that has sort of come in and is now getting distributed? And when I've looked kind of like at the genetics, I can explain most but not all of them. But again, when we look at the diversity, so here on the side are Klebsiella lineages, and these two genes we find in all of them. So this is the CTX, the ESBL gene, which is also what they were pre-selected for, so this is kind of like a self-fulfilling prophecy, I guess. And then here you see the resistance profile, and you can see obviously everything is resistant against cephalosporins because this is what the ESBL kind of like enzyme does and what they were pre-selected for. If we look for the alternatives, so what do you do when you have an ESBL organism? So there's either carbapenems, which is just a different class of beta-lactams, or you can combine cephalosporins with inhibitors. And so how that works is you treat the bacteria with the cephalosporin. They have an ESBL enzyme, but the inhibitor actually blocks the ESBL enzyme and therefore makes the cells basically susceptible again because the enzyme they use doesn't work anymore. And what we've seen here, and this is also part of what I'm going to follow up in my research more, is that for this inhibitor treatment, we get a very scattered picture. And it does not fully correlate. So there's some correlation with this enzyme NDM1, which is a carbapenemase, but there is not a complete explanation of why we see this scattered pattern. Where we see much more variation, and it's much less investigated, is the SHV enzyme. So this is a chromosomal enzyme Klebsiella does have. There are, however, also kind of like additional ones. So sometimes you see 2 per strain, so then it's plasmid acquired. And SHVs also, depending on what sort of mutations they have a broader or thin or smaller sort of like spectrum. So this might explain part of the variation, but again, not all of it. So there's kind of like more to be understood. So unfortunately, I will end quite a lot of things with kind of like questions we still have and not just yet the answers. And so what we've seen really from this kind of like comparatively tiny study to kind of like what's coming out nowadays. So we have several main lineages, so it was not one lineage which was floating around, which of course would have been an easier answer, but it's also what you don't see that kind of gives you the answer. There are definitely a lot of different resistance mechanisms, and so the NDM1 enzyme, which is kind of like targeting the last class of really well usable antimicrobials we have, at that time was introduced on different resistance elements and plasmids. And so the main lineage actually didn't carry this. So it's not that having the highest sort of like resistance repertoire makes you the most successful one necessarily. And again, kind of like the one which we've seen back then, so that was data from the 2000s, are not necessarily successful today. And the successful lineages, did not carry this gene. So it's not kind of like an equation that easy that, you know, the more resistance you have, the more kind of like you expand and the more successful you are in a clinical setting. And so, the resistance profile cannot explain this. And one of the problems that we've had kind of like is that we don't have a follow-up study. And so what we currently get from Klebsiella kind of like genomic studies is kind of like a study in this setting and then a study in that in a different setting. They usually have different patients, are in completely different locations, and are from different time frames. So it's very hard to see because, for example, we don't know if this lineage is actually still circulating there. So we really kind of like need more longitudinal pictures of the same setting, because at the moment it's really hard to compare neonate station with, for example, terminal cancer patients with kind of like patients in an environment like US, where of course the immune status is completely different. And so the thing, kind of like the advantage or what we can see in a longitudinal study, I'll just show you on the second example here. And this is kind of like the figure that kind of like inspired this study. And this was an observation by Matt Allington from Cambridge Uni, who is now at PHE. And this is when you look over a long time frame. So here from 2000 to 2012. And this is Enterobacter compared to Klebsiella. And what we can see in the different gray shades is the percentage of isolates resistant against classes of antimicrobials. So that's just a very crude way of kind of like grouping them, how resistant are they. And so in light gray you see kind of like only against one class, and then we go down to more resistant ones. And here for Enterobacter again, we only have ones which are already against kind of like 2 classes and then against 3. And what was interesting here is in 2007 there was a major change in the prescribing regimen to kind of like lower the amount of antimicrobials prescribed in the UK. And what you see is that in Klebsiella we have quite a clear kind of like reaction of the resistant population to this change, whereas in Enterobacter it wasn't really kind of like touched by that very strongly. And so the interest was kind of like to compare those two, because usually Enterobacteriaceae are all sort of lumped together a little bit. What the difference is and how does antimicrobial resistance actually, or changes in the treatment actually kind of like then, how are they reflected in the population and what makes them react differently. And so this is a slide which is summarizing a couple of years' work, I guess. This is the population of Klebsiella, and this is the population of Enterobacter from one hospital during this time frame. And it was kind of like sampled in a quite methodological way so that we're not biased towards outbreaks and so on. And what we've noticed is that when you look at Klebsiella at the time, then you can see an almost perfect replacement of the major lineages. So the 4 in blue are the main STs that we find. And you can see that they just replace each other in about 1.5 2 year time frames. And if we only look, of course, at like a 1 year or 6 month study, you never see kind of like these patterns. And the interesting thing is, so these lineages don't fully disappear. So it's not like one completely goes away. It just goes in the background again. And we don't know why, but this is kind of like one of the key interests. What is causing this kind of like replacement, and some lineages suddenly being better or more prevalent than others. And the opposite is happening in Enterobacter, where really we are sampling from the diversity all the time. So you can see kind of like all lineages are kind of like coming across throughout the years. And so obviously on a tree, you quite easily see kind of like patterns that you think you're seeing, but I've also done a statistical test. And so here you look kind of like, this is the growing distance over the tree. So these are very closely related isolates, and these are very kind of like far away from each other on the tree. And here you see the correlation with the temporal signal. So are they more prone to be in the same 2 year block or not, is sort of the test here. And you can see that in Klebsiella we have this really strong correlation where pairwise isolates that are very closely related, so from kind of like the same lineage, are very strongly likely to be in the same 2 year block. And if we go kind of like to the ones which are distantly related, they are more likely to be at different time points. Whereas for Enterobacter, this is kind of like there's no temporal signal at all. And I've also tested it with kind of like using months or years and trying to randomize the data. So we are quite certain of this signal. And this is quite bizarre. So it kind of like looks to me like a flu, for example, cycling ski trend. And the other thing that's different between those two bugs is when we look at the main STs again. So here we've just got the 4 major, so the most isolates STs for Klebsiella, and here the ones for Enterobacter. And the lighter color is the total number, and the darker color are the highly resistant ones. And you can see for Klebsiella, almost all of the main isolates from the main sequence types are also very resistant. Whereas in Enterobacter, if we look at the main sequence types, you can see that here we've got about half, and the others are not resistant. So there's no correlation in Enterobacter of how many we find to how resistant they are. And the other thing is, how well can we predict resistances based on the acquired resistances? So how much is dependent on plasmids and acquired genes? And if I look at the 3 major classes here in Klebsiella, again, I can explain almost all of the resistances, which kind of like correlates with the fact that Klebsiella is very dependent on mobile elements and acquired resistances. Whereas in Enterobacter, we've had this huge discrepancy in the beta-lactams, where we've seen just a couple with the acquired ESBL genes, whereas there's a huge number that is actually resistant. And so that is, of course, because Enterobacter has a chromosomally encoded AMP-C gene, which it can upregulate. And when it upregulates it, it becomes an ESBL phenotype. So this is why we see all these resistances without acquired genes. I've tried to map kind of like known mutations to, because this is quite a complex cycle, to these genes. And what is published in the literature in like biochemistry studies, it doesn't match at all, sort of. So if you look through these mutations, it really doesn't explain the phenotypes we see, which is probably because Enterobacter is a really diverse species. So these lineages are very, very different from each other. And so if you, for example, have a kind of like paper that shows a mutation here, works for example, an organism of this group, these ones might just be so different, and the interactions between these proteins be so different that you can't just map it across the whole diversity. So this is something we are planning to follow up in more detail with transcriptomics but that ended up being a bit too much for this paper. What we have seen is, when I looked then for kind of like the usual suspects, sort of like, so this is the CTX15, which is our usual kind of like resistance carrier for ESBL resistance, the TEM1. And then here we've got aminoglycoside resistance and another gene for aminoglycoside resistance, and in the context they usually appear with the kind of like transposable elements that mobilize them. And what we do see is that Enterobacter as well as Klebsiella have access to exactly the same pool, so they share the same pool of mobile elements. However, when we then look at the plasmids, and so here we've done PacBio of a couple of isolates that we've just selected from across the tree, you can see that every single Klebsiella and Enterobacter strain we've sampled has a completely different plasmid repertoire, even though their resistance cassettes are all the same. So we have a huge diversity here where the mobile elements are probably also moving between the plasmids and plasmids moving between the different lineages. But again, Enterobacter has access to sort of the same pool of plasmids as we can see in a few of them. But of course, because at the time ESBLs were kind of like the main treatment pressure on the bacteria, and it has an intrinsic system to get kind of like to avoid that, there doesn't seem to be a necessity to keep these plasmids. So there does seem to be some sort of cost, at least for Enterobacter, or some sort of like negative kind of like effect of carrying a plasmid. And so yeah, we have very different population structure and dynamics, and so all Enterobacteriaceae are not just the same, but the different species do actually need to be looked at kind of like separately, and you do need to take the biology a bit into account. We have seen this 2-year kind of like cycling period, which again kind of like emphasizes for me that we really have to look at longitudinal studies and to see kind of like what is a successful lineage in the hospital over a long time frame. And yeah, Enterobacter has the kind of like intrinsic resistance mechanism, thus a very complex small number of plasmids in our data set. And both have access to the same transposable cassette and to the same plasmids, but there's much more variety in the plasmids than in the actual cassette. So those seem to be far more stable, at least based on data we've got so far. And so yeah, this is really, I think, that the key point that I've kind of like hoping to bring home is that longitudinal studies are really important to see kind of like key shifts in the population, and that you need to look over long time frames. And yeah, we really need studies looking at all isolates, because a lot of data we have is very biased towards resistant isolates. And of course, if you only look at kind of like a part of the population, basically the tip of the iceberg, you're kind of like missing where they actually come from. So are they actually floating around before and then gradually pick up the resistances? Or do they just occur when they are resistant? And does this change more than just being antimicrobial resistant, but does this kind of like change something else that makes them more prevalent in hospital isolates? And yeah, can we kind of like, it's kind of like the final thought, I guess, can we exploit genomic insights when we look at these changes, when a new lineage kind of like becomes, like spreads more in the patients, to identify something that we can then target and make the bacteria actually unfit again. So this is kind of like, I guess, the actual outcome I would like to kind of like learn from all this. And so one of the things that I haven't mentioned, and that's because it's usually kind of like not really focused on, is intrinsic resistance. And so that means that it's not a gene that you acquire with a mobile element, but it's something in the chromosome, and you change kind of like usually the expression pattern, and thus become resistant. And so two kind of like classical ways to do this are efflux pumps, which are basically pumping things out of the cell in a way. And so if you have a lot of toxic material inside, you just pump out more. And the other one, which is more often used by Klebsiella, or at least more often studied, I guess, is if you deactivate the big porins, then you don't take up any antimicrobials and you don't have anything to worry about, in a very simplified kind of fashion. And we do know that it's important in Klebsiella. So there are studies where quite often it is either a switch from being ESBL, so cephalosporin resistant, to carbapenem resistant, or just to increase your resistance kind of like above the cutoff point. Why this isn't looked at so much? So we have a lot of really nice tools to search for acquired resistance. There are very good databases with all the acquired genes and so on. For intrinsic resistance, not so much. So I've thought to address that a little bit, and this is because I think this is really a neglected area of research. partly because it's a bit harder and kind of like less easy to pin down. You don't just acquire a gene and then you know you're resistant. And the problem is also it provides resistance against various things in one step, of course, because if you don't, it's quite, it doesn't target a certain class. And the other problem, and so one of the key things I'd like to is to find a way to sort of rescue the beta-lactams in the end, because I think this is one of the best classes of antimicrobials we really have. And there is currently a major initiative to find more inhibitors and kind of like combine beta-lactams with inhibitor treatments. And we also have inhibitors now for cephalosporin enzymes. So that means that you can add these inhibitors, they deactivate the enzymes, and your organism is basically susceptible again because it's lost the enzyme that does the job. But of course, if you stop any import, you also stop the import of the inhibitors, and all this kind of like effort is kind of like gone in vain, sort of. Of course, the advantage is this is messing up very basic cellular processes. You can't just stop all your porins because you have to get nutrients in somehow. And we don't really know what the effect of the fitness is. And so, so for Klebsiella, there is quite a bit of literature that they can deactivate the major porins. But of course, who then takes over the job? Because they do kind of like keep this mutation long term. So this is not kind of like a short reaction. And so because there isn't really anything to search for that, I've kind of like written my own sort of like workflow pipeline, I guess, which is hopefully going to appear on bioRxiv in the next week or 2 or 3. And so I've started with kind of like just using hammer libraries and basically my background together with Alex Bateman on what is usually sitting in the bacterial surface. And the idea is that you give it kind of like your genome set of, 300 Illumina genomes, and you get out kind of like the surface proteome, basically. So how many copies of which porin and efflux pump especially, but also, of course, everything else are there in the surface. I'm not going to go through the details, but we did kind of like play around quite a bit on getting this more or less as good as kind of like possible without doing everything manually. And so this is what you then end up with, basically, is kind of like here's your tree of isolates, and then here there's like a presence-absence matrix and the numbers, and of course you also get the sequences and everything, and then you can dig into more detail, for example, the phylogeny and which ones have expanded in which clade and so on. And so this is basically what this Klebsiella surface proteome looks like. So here I've just used Cat Holt's very kind of like basic global data set, which however does give us a very nice kind of like a comprehensive overview. So this is both the outer membrane, there just wasn't enough space. And what you can see is that there is a huge diversity of porins, of course, quite a lot of copies of some of them. There isn't very much else. So as I kind of like quickly mentioned at the start, part of my work is also to look for vaccine targets. So we found kind of like these guys here, the Fimbriae being pretty much the only thing in Klebsiella that's sticking outside, because that was one of the drivers to also write this, to look for vaccine targets, right? Because things that are sticking to the outside are usually based in the outer membrane through a beta-barrel structure, kind of like all of these. But we've seen this huge diversity of porins. And so, of course, if you inactivate, for example, your major porins, then you might just upregulate others who then take over the job, because before I starve, I'd rather use a suboptimal porin than kind of like go extinct. And so this is really kind of like work in progress, where I've kind of like just started to look at a few outbreak data or like clinically relevant data sets then. And this is again this Pakistan data set where I've just plotted kind of like 2 examples. So this is a family of efflux pumps where you see the different distribution. And then here, so the color change was possibly improvable because here green means present and red means deactivated, whereas here red is present. So apologies for that, but what you can see, and this is only the Lam B porin family. So this is only one family from this whole zoo of porins here. And here already we can see quite a bit of kind of inactivation and so on going on. And this is really one of the things I want to focus on more in the next couple of years, I guess. And so, this is kind of like the main kind of like question. So can we make successful lineages unfit again? Because I think it's not realistic that we will ever find anything nobody is resistant against. There will always be resistance mechanisms. But can we sort of like target some of them to actually be disadvantageous and not kind of like be selected for? And the other thing is that already, of course, we already have a lot of licensed drugs. So this is kind of like a way how to address this. Ideally is to find something that would actually, again, if you combine different drugs, exploit this and kind of like, for example, target that you would actually need to import more, but if you import more, you also import more of the other drugs, something kind of like along these thoughts. And yeah, I think what is really important is that we have longitudinal sampling and see kind of like major changes in the population structure, so that we actually see, first of all, which lineages do we need to focus on, or what makes them better at spreading in the hospital, because it's very clear what we've seen. It's not antimicrobial resistance as such, and it's not a certain plasmid strain combination as such. So in Klebsiella, we're a bit unlucky as compared to some other organisms where you can narrow it down much more on kind of like one lineage which has just expanded across, for example, sub-Saharan Africa in INTS, in Salmonella. And yeah, the idea is kind of like to target then these major adaptations and to kind of like stop these kind of like successful lineages being successful again. And kind of like at the moment I'm mostly working so I'm trying to combine kind of like my background knowledge and what I've just shown you on porins, for example, looking also on kind of like metabolic kind of like profiles, how they react to kind of like those different changes. So I'm not kind of like ignorant of kind of like machine learning and all the things that have happened recently. I think at the moment it's a bit tricky with Klebsiella, so it needs a bit more digging into detail, because quite often the main signal you get is the acquired resistance gene, and this is kind of like pretty much explaining it. So to look a bit more into the underlying things, but I am following it, and there's like programs coming out pretty much every other week these days. So there's a lot to kind of like try and implement and kind of like make more out of our data. And yeah, in the downstream, of course, so I mean, I'm a biologist at heart in the end, and I want to look at what is actually happening, and also all these predictions, kind of like how does this actually turn out, and what's the impact of changes just in the transcriptome, in the proteome, and also follow that up ideally with microscopy. For example, if we mess around with porins and efflux pumps, how does this actually change the import of beta-lactams? How many do actually end up going into the cell? And so yeah, this is kind of like, most of this work was really done at the Sanger in the past few years, so huge thanks to Nick Thompson and Gordon Dugan, in whose teams I was during that time, and who were super supportive and basically let me do what I want within reason. And then thanks to all sorts of collaborators, especially to Hassan, who kind of like really made this monster effort in the first study, and without whom we couldn't have any of these data sets that we're kind of like looking into. And yeah, thanks a lot to all of you for your attention.
Thank you. A wonderful talk, Eva. Thank you. One of the things that gram-negative bacteria use their porins for, as well as nutrient acquisition, is osmoregulation. There seemed to be quite a striking level of, you said, pseudogene formation, so inactive porins. Do you think this might make the bacteria less fit when they get exposed to high osmolarity conditions?
Yeah, so I've sort of like skimmed over some details with that. But yeah, the kind of like acquisition of ions or keeping the right balance, actually also for example, osmolarity is kind of like the big question, but also things like cofactors for enzymes, where you often need kind of like specific metal ions, is something I want to look into. So I think the whole metal biology is a huge point to be looked at in the bigger question of, in the end, can we make them less fit again? And yeah, including kind of like exposure to different salts, and how does it look like in the human versus is it just an adaptation to the environment, where Klebsiella does spend a lot of its time.
Thank you.
Thanks.
Okay. So I wanted to just pick up on a statement that you made earlier, which is you said that resistance doesn't explain the success of lineages over others. Do you mean, are you talking about specific resistance mechanisms, or are you talking about resistance in general?
I think in this case, resistance in general, at least acquired resistance here. And I mean, it surely does make them sort of like more successful, as in you get an antibiotic treatment, and you don't kind of like target the bacteria. So obviously against that, it does work. But on top of that, there is something else, because all these lineages have access to the same antimicrobials, but some of them you only see once or twice, and then they just, all of them in for example, the first study were pre-selected for already the resistance level, which was treatable at that time. And so there is a different kind of like underlying genetic factor that within this pool of already highly resistant ones leads to some to just occur, because in, for example, that study, all bacteria had access basically to the same patient population. They were kind of like exposed to the same environmental pressures. And so why some are so successful at quite literally maybe sticking around, which might actually be one of the reasons, which is kind of like that Klebsiella is very good at forming biofilms. But there is clearly something in addition sort of to just acquiring kind of like resistance plasmids that makes some lineages sort of like just better at being in people. It's possibly not the correct readout for success as such, because it's an environmental organism, but at least success is in our interest to kind of like, yeah.
Is much known about the costs of different resistance mechanisms or coming from different plasmids? Could that be a part of the turnover process?
No, so that's kind of like one of the key interests, and I guess also share with Adam Roberts quite strongly. There are experiments kind of like ongoing. It's currently at the stage where there are very focused experiments, also from kind of like groups in Sweden and so on sort of like the fitness costs, but then to translate that sort of like into a kind of like bigger clinical collection and so on. So we still kind of like miss quite a lot of biology in between. And sort of the costs of plasmids, or what kind of like how much are the transposable elements selfish, and how much do they contribute to the cell, or just like not go away again, we also don't understand very well. Klebsiella is definitely very good at keeping a lot of big plasmids. How happy it is with them in the end, kind of like in the environment, for example, when it has to compete against very kind of like different pressures, we don't know.
Maybe I can just follow up with another one there, actually. So when we're talking about a specific lineage, how much genetic variation do you see within them over time? When we're thinking about them changing over time, is there evidence that they could be mutating themselves over time, or is it mostly down to the swap over of plasmids?
Yeah, that's still quite a that's still quite a mess. Sure. So at the moment we're going by sequence type, which is an extremely crude. So the Klebsiella lineages are extremely deep branching lineages, so that happened a long time ago, and they're very different organisms from each other, which is a bit of a challenge, because so the differences we look at between different sequence types is a huge number of differences, and then the three we might be looking for sort of. There are definitely also differences within sequence types, and again, it's not always the same. So we have one example of a very clonal expansion that happened throughout the North and South America out of very kind of like reasons we don't know. Possibly kind of like the drugs that are being used there, kind of like it's quite, we don't know where this semi-geographical signal comes from, because there we have a clonal lineage of a clone called ST258 together with carbapenemase that is very successfully kind of like spread over the continent, and those are quite closely related, and there we can actually follow, for example, that, for example, is a candidate which has inactivated its major porins and kind of like become even more successful. Others, like, for example, the ST15, which I've shown in Southeast Asia, so between Pakistan and Nepal, there's a huge diversity also within the sequence type, and that's more the kind of like general picture of problematic STs we see in Klebsiella. So there is also quite a lot of diversity within the kind of like argument I've tried to make, or like I guess the point of having a longitudinal collection is at the moment it's quite hard to compare them, because they are from very different settings, so we don't know if the difference actually would kind of like be relevant, or how it would sort of like pan out in the end if it would be in the same setting. So that's why it's currently a bit tricky even to compare the kind of like Nepal and Pakistan sequence types and make conclusions about which, how, to interpret sort of like the diversity. So yeah.
So it does seem like a great deal of diversity. I suppose compared with the Salmonella world, you're seeing sort of thousands of SNP differences between your lineages, are you?
Yeah, so I mean, between different STs, I wouldn't even do a core mapping tree because it's just too diverse. So within, for example, this ST, which looked, for a Klebsiella word, pretty clonal, we've got about 100 or so. So it does, when you have one lineage in one setting, it does go down to you can actually look at SNP differences and so on. It kind of like then, so between sequence types, those are very different kind of like organisms. And it's also quite hard to say anything about the differences because they might have just chosen a slightly different, for example, metabolic kind of like set of genes for something, which may or may not make any difference actually.
So I'm delighted you're going on to do experiments with some of these isolates, but how are you, with so much diversity, how are you going to choose the best isolates to look at, which will have contrasting phenotypes, I suppose?
Yeah, I think that's the idea of kind of like having a big genomic data set and then sort of like picking the points where I really see a strong signal, for example, a new lineage which suddenly has kind of like expanded in our data set or which has taken over the resistant population, and then to focus sort of on that switch. Ideally that, because I appreciate, so obviously the plan is to do large-scale experiments, but the hope is with kind of like the genomic data as a sort of like starting point to filter out then key events that we want to investigate and pick kind of like that range of isolates where changes seem to have been kind of like crucial or beneficial.
Right. And one of the big struggles in big genome projects is getting hold of strains. Getting hold of DNA is one thing, but getting the strains. Do you think you're going to be able to access thousands of these strains? Yeah,
so I've got three very good, so I've got two very good collaboration and one kind of like publicly available collections which all span at least 12 and up to 20 years. So I'm very fortunate with my collaborators who actually stored all these isolates and are happy for me to access them.
That's great.
So that's kind of like an inclusion criterion for me at the moment, I guess, to look at these longitudinal collections only when I can actually then go back and kind of like dig into the details deeper with the isolates. Not necessarily having kind of like all 3,000 in my freezer, but the key ones that they are able to access.
Yeah. And it's great you come to LSTM to do this.
Yeah, thanks.
So you said that Klebsiella pneumoniae is an environmental organism. What evidence is there that the evolution and selection for resistance is actually happening out there in the environment rather than through people being prescribed antibiotics?
I mean, one thing is that we definitely see an effect of antimicrobial stewardship. So there is kind of like when you have a very kind of like so there's a study from Japan where they've made a nice comparison between three hospitals. One where cephalosporins were at the time kind of like the top sort of like resistance problem. And so one where the doctors could just prescribe them, one where they had to register that they are prescribing them, but nothing happened, and one where they actually had to justify the prescription. And you could see that the prescription rate as well as the resistance does go down. So there is an effect of, and of course all these kind of like studies only make sense in combination with infection control and with antimicrobial stewardship. So all this has to kind of like go together to actually have a final impact. The environmental pressure is a big unknown, but it's definitely something I also want to address. So together with kind of like people more in the environmental area who are more kind of like versed on that, because we don't know. We don't know the effect of, first of all, just pollution as such, which is like an obvious candidate. Pollution from antimicrobial factories is kind of a big problem that is kind of like known. But also other pollution, because of course we are firing at the isolates with, first of all, in the kind of like more in kind of like I guess high income settings with antimicrobials in pretty much every cleaning product you can think of and so on. So that's I'm absolutely convinced. And I mean, so Laura Piddock has shown some nice data that there is a very kind of like clear correlation to antimicrobial resistance. How much then kind of like other things, for example, like phage pressure or, you know, being eaten by kind of like protozoa outside and so on, how much that then actually also influences the population, which might be effects that we just don't think about at the moment, is pretty unknown. So there are ongoing studies, and so with Peter Taylor, we're also looking at kind of like phages targeting Klebsiella, and this is an ongoing theme in pretty much every Klebsiella lab at the moment. So we are kind of like, I guess, recognizing that there definitely is more out there, but I unfortunately, again, don't have a good answer just yet.
Okay, are there any more questions for Eva? Okay, we're all done then. I'd just like to say thank you again to Eva. That was a fantastic talk. I think it's a really important research area, and it's fantastic. It's really great to have Eva within the school and bringing a metagenomics expertise as well, which I'm sure many of us will be looking to collaborate with. So thanks again to Eva.
Thank you.